this last one? Or, I mean, grades at the bottom are pretty good. I could get a higher grade.
Okay, I'm recording. Okay, this is PS4. Our last workshop today. Questions 3, 4, and 5. So question 3, I probably will go over again because we just got what is it called? Dr. Spiro just reviewed it and said it was good to go. So I know I'm just gonna go over question 4 and 5 in depth, but like question 3, I'll go over again. I'll just post another recording for that. Okay, let me just minimize this. Okay, so this is question 3. The diagram illustrates a simple bacterial electron transport system in which formate HCOOH is used as the electron donor and nitrate is used as the electron acceptor. The two enzymes involved are formate dehydrogenase, FDN, and nitrate reductase, NAR. The periplasm is at the top and the cytoplasm is at the bottom. Note that some important details with which you should be familiar have been omitted from this diagram. So neither FDN nor NAR is a proton pump, yet electron transfer from formate to nitrate is accompanied by the formation of a proton gradient across the membrane. So our FDN we can see on this one, and then our NAR we can see on this one. So part A. Describe 2 ways in which a proton gradient is generated in the system. So the answer is usually going to be in black. It's going to be bolded. So neither of the protein complexes pumps protons. Thus, the first way a proton gradient is generated is through the proton's translocation that accompanies the reduction of Q and subsequent oxidation of QH2 that occurs across the membrane. Q cycle is not shown in the diagram that I just showed you guys in the previous slide. Secondly, there is a production of 2 protons in the periplasm, which is outside, and consumption of 2 protons in the cytoplasm, creating a proton gradient. And I'm just going to move on because. So this is basically our little equation, but I'll go more in depth when I record it again. Okay, so question B. For each molecule of formate that is oxidized by nitrate, how many protons are translocated across the membrane? Explain your answer briefly. So our answer here in bold is for each molecule of formate that is oxidized by nitrate, 4 protons are translocated across the membrane. Since there are 2 protons translocated by the redox loop, as well as 2 protons produced in the periplasm and consumed in the cytoplasm. Gonna move on to C. Okay, supposing FDN were to be located in the cytoplasm rather than the periplasm, what would your answer to question B now be? So I'm going to go back to question B. So it says for each molecule of formate that is oxidized by nitrate, how many protons are translocated across the membrane? Okay, so it's saying in the cytoplasm rather than the periplasm. So the answer to C is assume that the redox loop is still operating, so 2 protons are still being produced. But within the scenario, FDN produces 2 protons and nitrate reductive consumes 2. So these will cancel out, leading to a net translocation or transfer of only 2 protons via the redox loop for each molecule of formate that is oxidized by nitrate. Moving on to question 4. Okay, so Professor Timok Comet has developed a technique by which he's able to replace mitochondrial ATP synthase with a new enzyme capable of producing 1 ATP from 1 ADP and 1 inorganic phosphate for each 2 hydrogen ions, returning into the mitochondrial matrix from the intermembrane space. So 4A is if the pH inside the intermembrane space is becoming much lower than the pH inside the mitochondrial matrix, would you expect ATP production to increase or decrease? And then provide appropriate reasoning for your answer. So our answer is lower pH is an indication of higher H+ concentration, because remember, lower pH, the lower the pH, the greater the acidity. And the higher the pH, the greater the basicity. So that means the greater the higher the OH concentration when there's higher pH. Okay, so therefore there is a higher concentration of hydrogen ions pumped into the intermembrane space. With that in mind, more hydrogen ions would flow back through Dr. Comet's transmembrane protein into the mitochondrial matrix. By doing so, more ATP production is facilitated. Okay, I'm going to explain this answer more in the next slide. Okay, so the electron transport chain takes place in the inner mitochondrial membrane, and the citric acid cycle takes place in the mitochondrial matrix. So chemiosmotic gradient is just like how the osmotic gradient works. It flows from the high to low concentration. This is why the hydrogen ions are flowing from the inner membrane of mitochondria to the mitochondrial matrix like they were back here with Dr. Comet's transmembrane, because there was a higher concentration into the inner membrane space, so then it went back into the mitochondrial matrix. Any questions? Should I move on? I'm going to move on. So 4b, if a eukaryotic cell is utilizing Dr. Comet's new enzyme, how many molecules of ATP, net amount of ATP, or its high energy equivalents will be produced for 3 molecules of glucose undergoing glycolysis, Krebs cycle, and ETC in abundance of oxygen? So this question will be, I think, a lot of math. We'll move on. So a eukaryotic cell produces a net amount of 2 ATPs per each glucose in glycolysis. Therefore, we will have a net production of 6 ATP for 3 molecules of glucose via glycolysis alone. 3 rounds of glycolysis will also give us 6 molecules of NADH and 6 pyruvates. I'll break this down more in the next slide to make more sense. Okay, so this is part of our answer. It's not the whole answer. So 1 round of glycolysis, this is our whole glycolytic cycle. So in 1 round of glycolysis, there's 4 ATP produced, as you can see here at the bottom. 2 ATP consumed, 1 ATP here, and the second ATP here that's consumed. So we do 4 ATP minus the 2 ATP consumed, and we get 2 ATP. And then in this whole cycle, there's going to be 2 NADH produced and 2 pyruvates produced. And then it starts off with 1 glucose as shown in the picture. So the second one is 6 pyruvates. So this is still like the answer to that question, the ones that are in bold. 6 pyruvates are eventually converted to acetyl-CoA and sent into the Krebs cycle, initiating 6 rounds of the cycle. Each round of pyruvate acetyl-CoA produces 1 NADH, leading to the net gain of 6 NADH in the mitochondria. Each round of the Krebs cycle, or the citric acid cycle, produces 1 GTP, 3 NADH, and 1 FADH2. Therefore, after 6 rounds, we will have 6 GTP, which is equivalent to ATP, 18 NADH, and 6 FADH2. I'm going to move on. Okay. So this is basically how our glycolytic cycle will go. It's going to convert from pyruvate to acetyl-CoA, and that's going to go into the citric acid cycle. So each round of this conversion goes to produce 1 NADH. So when pyruvate goes to acetyl-CoA, we can see that there's 1 NADH produced. And then it leads to the net gain of 6 NADH in the mitochondria, because remember, we're going through 3 cycles. And then each round, so 1 round produces, of the citric acid cycle, produces 1 GTP, 3 NADH, and 1 FADH2. So these are basically with the glycolytic cycle and the citric acid cycle combined, the products that you get. after 3 cycles of glycolysis and citric acid cycle. Okay. Okay, this is still part of 4B's answer. So in total, we have 6 net ATP and 6 NADH in the cytosol from glycolysis, and also 6 GTP, which is also ATP equivalent. So 18 NADH and 6 FADH2 from the Krebs cycle in the mitochondrial matrix. Each NADH facilitates the pumping of 10 hydrogen ions into the intermembrane space, while each FADH2 only leads to the pumping of 6 hydrogens. Dr. Comet's enzyme facilitates the production of 1 ATP for each 2 hydrogens returning. Move on to, that just happens when it gets dark. Okay, so 24 NADH times 10 is going to be 240 hydrogen ions plus 240 hydrogen ions divided by 2, which is equal to 120 ATP. So this is still part of your answer because it's bolded. And then the reason why it's 120 ATP and not like 240 is because remember I said that there's 2 hydrogen ions that equal to 1 ATP. So that's why it's 120 ATP. So 6 FADH2 multiplied by 6 equals to 36 hydrogen ions plus 36 hydrogen ions divided by 2 equals to 18 ATP. And the reason why it's not equal to 36 is because, as I said before, there's 2 hydrogen ions that equal to 1 ATP. And then the rest is just as it was, 6 GTP from the pyruvate to acetyl-CoA reaction, 6 ATP from the glycolytic reaction, and then you just add them all together and you subtract the 6 ATP for transport of 6 cytosolic NADH from glycolysis because they're consumed, remember? Because there was 3 glycolytic cycles that occurred and 2 ATP consumed in each of the glycolytic cycle. So you do 3 glycolytic cycles multiplied by the 2 ATP consumed. And then you get negative 6 ATP. So 120, this is positive, plus 18 plus 6. Here, 120 ATP, 18 ATP, plus 6 GTP, plus 6 GTP again, plus 6 ATP minus 6 ATP equals to 150 ATP for net gain. So that's your answer. I'm going to move on if no one has questions. Okay, this is what I said before, that remember 1 ATP is equal to 2 hydrogen ions, so like 240H plus is actually equal to 120 ATP, 36H plus is actually equal to 18 ATP, just divided by 2. Okay. Question 5A. These problems relate to bacteria. Remember that bacteria does not need to export ATP, so there is no need to account for the energetic cost of export. In a sample of bacteria, there are 18 carbon units to its ATP synthase for each 2 electrons that enter electron transport from NADH. 2.5 molecules of ATP are made. For each NADH that is oxidized, how many protons are translocated across the cytoplasmic membrane? I'll just wait a little. So the answer to 5A is 1 rotation of the ATP synthase generates 3 ATPs. So each carbon subunit corresponds to 1 proton that will cross the cytoplasmic membrane. There are 18 carbon subunits, so 18 protons will cross the membrane per 1 spin of the ATP synthase, generating 3 ATP. We need to determine how many protons needs to cross the membrane to generate 2.5 ATP from NADH oxidation. So this is kind of straightforward. It's just like dimensional analysis. So we can set up 18 protons divided by 3 ATP is equal to X protons divided by 2.5 ATP. So when we solve for x, we get 15 protons translocated across the membrane per generation of 2.5 ATP. Make sure you write your units. These are your units. The protons translocated across the membrane per generation of the ATP on your problem set. Okay. Does that make sense to everyone? This problem set is really like mathy. Okay, so 5b, the oxidation of succinate in E. coli is accompanied by the translocation of 6 protons across the cytoplasmic membrane. If 1.5 ADP molecules are phosphorylated for each 2 electrons originating from succinate, how many C subunits does the ATP synthase have? I'm going to move on to the next answer. So this is just like the first one. So each spin of the ATP synthase makes 3 ATP. 1.5 ATP is generated per 6 protons crossing the cytoplasmic membrane. So per 1 spin of the ATP synthase generating 3 ATP, there must be 12 protons crossing the membrane. Each C subunit corresponds to 1 proton that crosses the membrane, so there must be 12 carbon subunits. So in other words, just to make it more simple, I just added dimensional analysis. You just do 3 ATP over 1.5 ATP times 6, and you get 12 protons. So pretty straightforward. Does anyone have any questions? Should I move on? Okay, I'm going to move on. I think that was it. Yep. Okay, so we're good to go. I'll go back over question 3. Your problem set's due on Monday? Okay, so I'll do it over the weekend. And I think that's it. This was our last workshop. I'm gonna post a recording today. So you guys are good to go.